Alright, so this lecture is going to go over chiefly on skeletal muscles. We do have three types of muscles. We got our skeletal muscles, we got our cardiac muscles, and our smooth muscle. We're going to focus mainly on skeletal muscle, and then at the end we'll touch briefly on cardiac muscle and smooth muscle. And those last two, cardiac and smooth muscle, we'll revisit again in the digestive system and in the cardiac system. Okay, but our chief focus here is skeletal muscle and the muscle physiology. We'll look at some of the differences between the three and relate some of them, and then again we'll touch on the others later on in the semester. All right, so here is looking at the three different types of muscles. We got our skeletal muscle, our cardiac muscle, and our smooth muscles. Again, our focus is going to be on skeletal, but here we can look at some of the similarities and differences between these muscle types. And what you can see between the skeletal muscle and the cardiac muscles is they have these striations. Okay, those striations are set up because those muscles each have sarcomeres, an arrangement of the contractile proteins in these muscles. They are going to be attached to bones for skeletal muscle, and cardiac muscles, they are going to be what cause a contraction in the heart. But you can see the skeletal muscles are these long cylindrical muscles. Okay, they can be 3 feet in shape, or 3 feet in length on some of the longer muscles, and they are multinucleated. They originally were single cells that merged or fused together, and so we have multinuclei in these skeletal muscles. The cardiac muscles, similar in appearance in that they have the striations, but they are these branched, smaller cells that are brought together by these little intercalated discs. And then the oddball of the bunch, the smooth muscle, you can see there are no striations, so they're not set up in these sarcomeres. And we'll take a look at this at the very end. We'll look at the arrangement of the smooth muscles. They give it this kind of smooth appearance. But where we're going to work with these are going to be like digestive tract, the muscles that control blood vessel diameters are where we'll find the smooth muscle. And of course, I didn't mention, but cardiac muscles we'll find in the heart for contraction. Okay, where skeletal muscles are going to be attached to the bones and allow for movement. Okay, we will see these. We're going to be looking at these chiefly, and so we'll look at how we produce movement by contracting our skeletal muscles. Again, chiefly we're going to be looking at skeletal muscles, so there is some terminology that is specific to these muscles. Muscle cells, you'll hear a term muscle fibers, so if you hear muscle fiber, that's an individual muscle cell. The cell membrane is termed sarcolemma, the cytoplasm, sarcoplasm, and then this specialized endoplasmic reticulum in the muscle is called the sarcoplasmic reticulum. We're going to see this is a place in which we have stores of calcium, because calcium is going to be important in the producing the contraction in our skeletal muscle, in our muscle fibers. So looking at our skeletal muscle as a whole, how is it set up, how is it arranged in the body? What we're doing is we're producing these levers by attaching the muscles to the bones and going across a joint. We're producing levers with the fulcrum at the joint, right? And the muscle can only shorten, and so this makes it so the muscle can only do a pulling force. There is no pushing force in the muscle. So the only force that can be produced by this muscle, say our biceps, is shortened, is it can pull this towards there. There is no force produced when the muscle is lengthening. Okay, the force comes from the muscle shortening. Right? And so this arrangement makes it so we have to have antagonistic muscles. When I do flexion of my arm, I'm using my biceps, but there's no force when I lengthen this bicep. There's no force being produced. So how do I get to do extension? Is I have the antagonistic muscle, in this case the triceps, that produce the pulling force to pull my arm into extension. Okay, so that is why we have to have these antagonistic muscles, because a force is only a pulling force. There is no pushing force to these muscles. It is the shortening that is causing the force. Now let's look at the structure of a whole muscle. So muscles are muscles that we think of are basically consisting of many different muscle fibers. It's not one individual big fiber. There are multiple muscle cells that make up the whole muscle. And so when we look at a whole muscle, if we cut into a whole muscle, you're going to see the little red dots here are the muscle fibers. I'm going to get my big head out of the way here. And so we have these individual muscle fibers, and these muscle fibers are going to be grouped, packaged together in what we call a fascicle. It's a bundle of fibers. And to create the whole muscle is multiple fascicles are grouped together to form this whole muscle. Okay. And in this muscle there is connective tissue. Our individual muscle fibers are wrapped with what's called endomysium, M-I-S-I-U-M, endo. The fascicles are wrapped by perimysium, and then the whole muscle is wrapped by epimysium. And so that is the arrangement. There is both a connective tissue component of the muscle, and there is a contractile component. The muscle fibers have the contractile elements as well as some connective. So there is both connective tissue and contractile elements that produce our muscle. Now diving deeper into the structure of the muscle, this is a cut of an individual muscle fiber. So one cell we're looking at here, and you can see that this muscle fiber cell has these multiple cylindrical structures here. These here, and there are multiple of them, these are the myofibrils. This makes up the main interior of the muscle fiber. And what is housed here are the contractile components of the muscle. Our contractile components are going to be made up of these thin and thick filaments, and basically the myofibrils are just sequence of these, what we call sarcomere, these arrangements of the thick and thin filaments down the length of the muscle. Right. Also surrounding these myofibrils is this blue structure, kind of this sheathing around it. That is the sarcoplasmic reticulum. That modified endoplasmic reticulum, that's going to house our calcium. Okay, as we said, the calcium is a very important component to induce our muscle contraction. So these myofibrils will run the length of the muscle. And you can see here is, say, one sarcomere there. And basically there'll be a sequence of these sarcomeres running the length of the whole muscle. And they're going to be surrounded by this sarcoplasmic reticulum. And the other thing to notice on the slide is these little yellow structure. Okay? Because what we're going to see in our muscle is similar to our neuron in that the neuron can be excited and we can transduce an action potential down the plasma membrane. We can do the same with the muscle. We can cause the muscle to get activated and send that action potential down the membrane of the muscle fiber of the sarcolemma. The key is that action potential is the key to cueing contraction. And to cue the contraction is this action potential has to have in vicinity of these parts, these arrangements of the sarcoplasmic reticulum. So you see these little yellow structures, they're almost like little tunnels. They're invaginations of the plasma membrane, and they're coming in from this pore here. All right, and the key of these, what we call T-tubules, is to bring the action potential into the inner portions of the muscle fiber. Okay, because the action potential has to get access to these sarcoplasmic reticulums. Okay, this is going to be key to causing contraction. So as that action potential is running down, we can move the action potential deep through the T-tubules in association with these deeper myofibril and sarcoplasmin arrangements in the cell. So looking closer at these T-tubules, these transverse tubules, you can see the arrangement. Here is the sarcolemma, in which the action potential would be running down. And now we can move the action potential deeper into the muscle in association with these terminal cisternae, so we can induce the contraction. Okay, we'll see this action potential actually induces the release of the calcium from the sarcoplasmic reticulum. And we'll see why this is important in our contraction in just a bit here. Okay, but again, the T-tubule's function is to bring the action potential deeper into association with the deeper T-tubules, sarcoplasmic reticulum, in association with what we call the terminal cisternae of the sarcoplasmic reticulum. Right? You might hear a term triad. Triad is an arrangement because you'll see the T-tubules are flanked on each side by a terminal cisternae of two different sarcoplasmic reticulum, one here and one here. Now here, looking closer again at the sarcoplasmic reticulum, these are those terminal cisternae. Get my head out of the way here. Terminal cisternae. And so these flank on each side of those T-tubules, right? And what's here is going to be those stores. They sequester the calcium. Okay, the calcium's here, and it's going to be released out into muscle, into the sarcoplasm. Okay, sarcoplasm is the cytoplasm of the cells, right? It gets released, so it has access to behind here those contractile proteins, those myosin and actins we'll be talking about here briefly. Okay, but this is where the calcium that is going to be involved in muscle contraction, this is where it comes from. It's released here, and then the calcium gets brought back in to cue muscle contraction, then muscle relaxation. So let's look at those myofibrils. Those are rod-like structures running down the muscle fibers. And what we're going to find in those myofibrils are the contractile elements of the muscle. We're going to find there's these arrangements of what's called a sarcomere in here, and this is arrangement of these thick filaments and thin filaments, and it's going to be the interaction between these two filaments that are actually going to cause the shortening of the muscle. All right, the thick filaments you can see are set up with these myosin heads, myosin molecules that have this head region, and then the actin, the thin filaments are made up of these actin molecules. All right, and they're set up in this arrangement in the sarcomere, and what we have is a repeating pattern of these sarcomeres down the length of the myofibril. All right, so there's a sarcomere. Let me get my head in here. There's a sarcomere and then another sarcomere, and so there's a series of sarcomeres all the way down the muscle fiber. And what we're going to see, what's going to happen with these is we're able to move these filaments this way and this filament in this direction, and when we do that, we're going to get shortening. All right, so in the next slides, we'll look at all these structures in detail and put it all together on how we're going to get this movement of the sarcomere to be able to shorten it, shorten the length, and therefore causing contraction of the muscle. Right, and so this myofibril proteins, there are multiple proteins here. The ones involved in contraction are the myosin here and the actin. Those are what make up the thick and the thin filament. Okay. What's going to happen is these heads, you can see these myosin heads, they have this tail along with this hinge region and then the heads, and that allows the myosin heads to pivot. So we're going to get the head, here's the head, and then here's the hinge region. We get pivoting. All right, and these myosin heads are going to interact with these G-actin molecules, these little balls that make up the thin filaments. All right, there's this yellow structure here, yellow portion of the actin, in which myosin will be able to interact, bind to, and then it's going to pivot. And what's going to happen is you can see if that myosin head binds here and pivots, it's going to pull this actin in this direction. And these heads are arranged in the opposite direction, and they're going to bind to actin and pull. So these heads will pull this actin in this direction. So you can see we're going to get the shortening of the sarcomere. Okay, four other proteins here, but let's first look at these two. These regulatory proteins are this troponin, this little brown protein here, and this green string-like protein is tropomyosin. And what you see taking place here is the yellow region, the region in which the myosin heads will bind to, is covered by tropomyosin. Tropomyosin is associated with troponin, and what we're going to see is troponin binds to calcium. All right, so when calcium's not present, it's not bound to troponin. Tropomyosin is sitting over the regions in which the myosin heads would bind to. And so if tropomyosin is in the way, the myosin head cannot bind to actin. So we can't get this interaction. We can't get what we call cross-bridging between actin and myosin. All right, if we can't get the cross-bridging, we can't get them to move the filaments, and therefore we wouldn't get shortening. That's when your muscle is relaxed, when we can't get this interaction. What happens when calcium is available is calcium will bind to troponin, and when calcium is bind to troponin, it will move tropomyosin away, and now with tropomyosin moved, my myosin head can bind to actin, and we can get interaction, and we can get contraction. Right? So basically these proteins regulate whether we're going to get contraction, whether muscle is contracted or relaxed. All right? And the cue for these to operate is going to be that calcium binding to a troponin. So that's where our calcium is going to come into play. Two other molecules here. We have this long tentin. This is one of the longest proteins in the body. Largest protein, tentin. It basically helps anchor, support these thick filaments. Right? And then we have nebulin. You can see nebulin down here. It helps. It is the structure down in the between the balls, between the G-actin molecules, and that helps to anchor the actin or the thin filaments here. So looking a little closer at the thick filaments, as we said, they are made up of multiple molecules, myosin molecules, with these heads arranged in this direction. The heads pivoting this way, and these pivoting this way. And remember, these heads, or this myosin molecule, has a hinge region. So that's going to give us our pivoting of the molecule. What else? This pivoting taking place, all right, we're creating movement. What do we usually need to do if we're creating some kind of movement in the cell? We're going to need some form of energy to make this, what we call the power stroke, having the myosin head move on that hinge region. So we're going to need ATP. All right, and we need the ability to cleave ATP, and so these myosin molecules have ATP activity. Cleave ATP so they can use it to do the power stroke, to torque on that hinge region. All right, there are different forms, isoforms, different forms of ATP activity, and that comes into play when we have fast twitch and slow twitch, and this slow twitch, but it's more like a medium twitch in between the two. And these different isoforms of ATPase give us the speed in which we can move that head. All right, a fast twitch muscle going to have a faster ATP activity, and we're going to be able to move that molecule quicker. Where the slow twitch, they won't move as quickly because we can't cleave the ATP as quickly and the movement, the power stroke is slower, and therefore it's slower to contract. Our fast twitch very quick to contract, our slow twitch not as fast. Okay, so that's going to make up those thick filaments. We're going to have those heads, and remember, these heads are, this region is what's going to interact with the actin. So we get binding of the actin, myosin to actin, pull, myosin head's going to let go and say there's another molecule down here. Myosin's going to bind, pull that. So we just keep pulling sequential molecules of actin with the myosin heads, right? And that is going to allow us to do our shortening. All right, and then finally we have some of the accessory proteins. That is the tentin, all right, and the nebulin. The tentin is a big, long, elastic molecule here. This green is one of the largest proteins in the body. As you can see, elastic allows a stretch of that sarcomere, so that's part of that elastic component of the muscle, along with the connective tissue, but it also anchors in those thick filaments into the sarcomere. Right, and then the other we saw earlier was the nebulin. and that is that little blue that runs inside those twisted, beaded chains of the thin filaments. It attaches to the Z disc and anchors in those thin filaments. It is inelastic, so it doesn't have elastic properties like the nebulin does. So before we look at the elements that give us contraction, let's first look at the sarcomere and what happens to the sarcomere when contraction takes place. All right, this is the functional unit of the muscle, the sarcomeres, because this is what's going to shorten and cause our contraction and create our tension or create our force by our muscles. All right, so there are different regions. You can see here. Here's a photomicrograph. Hopefully that looks familiar to you from the anatomy days. But if you remember, there were these dark lines and there's these striations. Okay, this is looking in very close. You can see a little more details. You probably just saw light and dark striations with the microscopes in anatomy, depending on how powerful they were. But on each end of the sarcomere is the Z disc. Okay, that's these here. There's the Z disc. All right, they flank both sides, and they're going to be basically kind of those anchors for the thin filaments, the site of attachments for them. There is that light region. Okay, this is here. This is the I band. What the I band contains is only thin filaments. Okay, it's the region that has only thin filaments only. Okay. And then the A band, that's the darkest region of the sarcomere. That's this here. Right? The A band spans the length of the thick filament. But remember, there's some overlapping thin filaments near the thick filaments. All right, so the A band encompasses the entire length of the thick filaments, but there are regions that have thick and thin. And we're going to see there is this H zone. The next line, that is a little lighter region. We'll see that H zone. Okay, the H zone is the central region of the A band, and it contains only the thick filaments. So both the I band and the H zone are H zone's only thick, I band's only thin, and then in the middle of that H zone is the M line, and that forms the attachments for the thick filament. All right, so that is the setup. Those are the different regions of the sarcomere, and we're going to see there are some changes that take place to these when we actually get contraction of the muscle. And so we've been looking at this sarcomere in a two-dimensional type structure, but what we see is it actually, remember, it is in this rod-like structure. And that rod-like structure has multiple thick filaments and thin filaments, and you can see there are roughly 3 thick around each thin, and there's 6 thin filaments that encircle thick. So we have not just this two-dimensional structure, but it is this combination, this round cylindrical-like structure in which we get these will be binding to those regions of the, and we get the shortening down. Okay. So how does this all work? How does this shortening take place? That's where the sliding filament theory comes into play. Right? The sliding filament theory states that the filaments are sliding across one another. The filaments are not changing in size. The thin filament's not getting smaller. The thick filament's not getting smaller to cause contraction. Basically, we're just having those thin filaments sliding across thick. Therefore, sliding filament theory. They are fixed length. So you can see what's happening looking at our sarcomere when our muscle is relaxed. When contraction is not taking place, you can see there is overlap between the thin and thick. You can see the H zone. The H zone is that thick filament only. The I band is thin filament. And so when these filaments slide across, when those myosin heads are pulling the thin filaments towards one another on each end of the sarcomere, what happens is the thick starts ingressing into the I band. So the I band, you can see, gets smaller, shorter, and the H zone, where there was only thick filament, now the thin filament is encroaching into that I zone as it slides, and we get the H zone also getting smaller compared to what we see in relax. Okay, so that is how we're going to shorten. Remember, we have a sequential running down. We have, here's one sarcomere, another sarcomere. So we have this repeating pattern of sarcomeres down those myofibrils, and when they all shorten, that muscle is going to shorten. So we now have seen the structure of the sarcomere and kind of how contraction is going to take place, but let's look at the steps that will get us to contraction and then relaxation of the muscle. There are three main steps there. First, we have to get the muscle excited. Get the muscle excited, the motor neuron has to talk to the muscle. Our muscles are consciously controlled. We have neurons in the brain, that pathways that send down to our muscles, and so that muscle, so the events at the neuromuscular junction where the motor neuron and the muscle meet up, we have those events. That's where we're going to get the muscle excited. Remember, the muscle is like a neuron. It has to be excited, and then we start the action potential on the muscle. The steps between going from action potential to getting into the actual contraction is called the excitation contraction coupling. This is, or you'll hear it abbreviated as EC coupling. So EC, excitation and contraction. All right, these are the events that take place to get us to contraction. If you remember from the beginning, we were talking about calcium. We have those calcium stores in the sarcoplasmic reticulum. We got to get the calcium out because the calcium binds to that troponin, moves the tropomyosin, and now we can get contraction. All right, so these events that are taking place at the neuromuscular junction and the EC coupling are essentially there to get that calcium out from the sarcoplasmic reticulum. Okay, so we'll discuss these events, but let's first look at the last event. We'll go in a little bit backwards order. We're going to talk contraction and relaxation of the muscles first to get us our muscle contraction. All right, and to cue this is we have to have that calcium signal. Okay, we're going to need two main molecules to make this take place. We're going to need calcium, right? We get that from the sarcoplasmic reticulum, and we need ATP because we got to have energy to be able to cause those myosin heads to pivot, to do what's called the power stroke, to be able to pull those actins. All right, so we'll go through these three steps, but first we're going to head off. We're going to go a little backwards and go in contraction and relaxation, and then we'll head back to getting that calcium signal out through the events at the neuromuscular junction and the EC coupling events. All right, so calcium. What is calcium for? We've already seen a little bit on when calcium is not available. It is not binding to troponin here. And when troponin is not bound to calcium, this tropomyosin, gray-like protein, is covering the region in which myosin will interact with actin. So when that is covered, the myosin can't interact. If we can't get cross-bridging and power stroking, pulling of the actin, we don't get contraction. So when the muscle is relaxed, when calcium is not available, myosin and actin cannot interact. All right. Now we get our calcium. We'll talk about how we get it, but we get our calcium from our sarcoplasmic reticulum. We release calcium from the sarcoplasmic reticulum, binds troponin. Troponin moves tropomyosin, and now we can get that interaction of the myosin and actin head, and we'll see we can get what's called the power stroke. And you can see here we're using ATP to make that power stroke happen. And that's all we're going to be doing. As long as calcium is available, myosin can interact. This head's going to release, bind to this one, pull this way, release, bind to this one, and pull this way. Okay, so that is how we're going to get the movement. How we're going to slide those filaments is by pulling them with our myosin head. So now let's look at how this contractile cycle takes place, how the myosin head binds, power strokes, and then detaches. So there's a state called rigor where there's no ATP bound. The myosin has already gone through what's called the power stroke, and there's no ATP bound. There is a state of rigor where the myosin head is immovable. It's attached to the actin. In a living cell, this rigor state is very brief because we have ATP available, and ATP is going to allow us to release from the myosin or from the actin. Okay, myosin will then bind again, and we'll see a power stroke. So we'll see how this in this next slides, how this using of the ATP to allow for the power stroke of the myosin head. All right, so I'm going to skip a little bit further. You can read these, but let's look at this next slide showing how this power stroke takes place. All right, after a cycle. So basically all we're doing with the myosin is we're binding, power stroking, releasing, going up, binding a new molecule of actin, power stroking, and releasing. So how does each of these cycles take place? This is just one cycle of this contractile cycle of the myosin head. So after we've used up the ATP to do a power stroke, the myosin head, so we'll see the myosin head can be at 45 degrees when it binds, and then it turns to 90 degrees to pull the actin. After one contractile cycle, the myosin has used up the ATP, but it is still bound to actin. This is that rigor state. In a living cell, it's not rigor mortis, because in a living cell we have ATP available, and so when ATP binds to that myosin that's already done the power stroke, that's in rigor, the ATP binds, and now myosin will release. Let's go down here. The myosin head hydrolyzes ATP, and the head rotates. You can see here, when it binds ATP, it releases, rotates back to that 45 degrees, and it will bind. It cleaves ATP to ADP and phosphate, and it binds to the actin. Right, to do the power stroke, we release the phosphate. That gives us our power stroke, and then the ADP is released afterwards. Now we're no ATP, we're stuck in that rigor state, and we just begin again. We bind a new molecule of ATP, we pivot back, bind to actin, and do this. So we just keep doing this as long as ATP is available and as long as calcium is available. Okay, because remember, we have to have calcium to get the interaction between myosin and actin. Right? If we don't have calcium, remember that tropomyosin is in the way and we can't get the interaction here. Okay, so how it works is, say we already did in one power stroke, there's no ATP. All right, ATP binds to my myosin, that releases, and the myosin goes to 45 degrees and will then bind to a new molecule of actin. Right, ATP is cleaved to ADP and phosphate. The phosphate's released, we get the power stroke, and then ADP is released. We're stuck again, and we're back at that cycle. So that is, and we're just continually doing this. So you can see how energetic a muscle is, because we have all these myosin heads, and each time we're pulling the actin, we're using up a molecule of ATP. So you can see how much ATP we got to produce to be able to cause these muscle contractions. All right, so we've done those contraction events. All right, we'll hold off on relaxation in just a bit, but we've seen what needs to take place for contraction. Right, we have to get that calcium out, and then we get the events, we get the interactions between myosin and actin, and we get the power stroking, and we get that sliding filament theory. Okay, now let's see what are the steps to get this calcium out? How do we control? Because that's going to be the switch to turn the muscle on for contraction or relaxation. It's getting calcium into the muscle, into the sarcoplasm, because it is in the muscle already. It's in those sarcoplasmic reticulum, but to get it into the cytoplasm of the muscle where it can interact with myosin and actin, and then how we remove calcium. Okay, to do that, our first is we have to get the muscle excited. That's going to take place, those are the events that take place at the neuromuscular junction, where we have the muscle and we have the neuron coming in to innervate. Right? We have the little synapse with the muscle, and that forms the neuromuscular junction, the synapse between the motor neuron and the membrane of the muscle fiber. So here's that setup. Here is the neuron coming in and talking to the muscle. This is the neuromuscular junction form here. All right, let me get my big head out of the way, and we'll look at a little bit closer setup. And what we have here is, here's the neuron with vesicles, and our neurotransmitter is going to be acetylcholine. The receptors are nicotinic acetylcholine receptors. And what nicotinics are ligand-gated ion channels. It's going to allow for our sodium. So when acetylcholine is released by the motor neuron, it binds to the nicotinic receptor, now sodium can rush in, and that's going to cause depolarization of the muscle fiber. That's going to give us our excitation, right? Just like our neurons, when we get excited, the muscle fibers, we have to get excited because we're going to be able to hit threshold just like a neuron and cause an action potential on the muscle fiber. Right? But at the neuromuscular junction, what we're getting is we're getting the excitation. We're getting the depolarization. We got to get enough depolarization to initiate that action potential. So the steps involved here the action potential driving down. Hopefully by now you guys realize we got the calcium, that action potential cues those voltage gated calcium channels open. Calcium rushes in, activates those snare proteins to cause the excitosis of acetylcholine. Now we get the neurotransmitter bound to our nicotinic receptors, and say our sodium is out here. All right, our sodium now can rush in, and we're going to get depolarization of the muscle fiber here. We're going to get enough depolarization to get the threshold. Remember, we're going to do an action potential, so we have voltage gated sodium channels that get cued from the depolarization caused by the motor neuron talking here. Okay, the acetylcholine binds, and then we have our acetylcholinesterase like we've seen before, and breaking down acetylcholine because we don't want to have the muscle just always contract. We want to be able to release acetylcholine, get the muscle to contract, and then we can relax it. Right? Stop cueing this event and the muscle relaxes. There's the events. Here's a little bit even closer broken down is the events that take place at the nicotinic receptor. It allows potassium and sodium in, but the more influx is sodium, and therefore we get depolarization by opening of these nicotinic receptors. Alright, so once we have those events taking place at the neuromuscular junction, we get our excitation, we get the depolarization of the muscle there at the neuromuscular junction. And what do we have there? We have those voltage gated sodium channels. So now we're going to induce that action potential that can run down the sarcolemma, run down the plasma membrane of the muscle fiber, very similar to what we see in the neuron. All right, so that brings us to the excitation contraction coupling phase. Right, this elicits excitation. This here, we got the excitation. Now we start the action potential. Action potentials running down the sarcolemma is the beginning of our EC coupling, excitation contraction EC coupling phase. Right, and what's that going to lead to? That's going to lead to us getting the calcium signal out. Right, so all the events from action potential to getting calcium out till we actually start contraction is the EC coupling phase. Let's look at what's taking place. Here are the events at the neuromuscular junction. Okay, we have that nicotinic receptor causing activation. We have the sodium influx, and that's going to induce the action potential. Now we have the action. So we got our muscle excited. So now we have the action potential. Starts our EC coupling phase. And the action potential is going to run down those T-tubules, right, that are flanked on both sides by terminal cisternase of the sarcoplasmic reticulum. Down these T-tubules, we have these DHP receptors, dihydropyridine receptors, on the T-tubules. Okay, this is a calcium channel. It is coupled to these ryanodine receptors on the sarcoplasmic reticulum. So what is happening is that action potential is going to cause a change in the DHP receptor that is coupled to the ryanodine, and that's going to cause a change to open up the calcium channels on the sarcoplasmic reticulum. Remember, we have these large stores of calcium. Calcium is what we need to induce to bind to troponin, to remove trypomyosin so we can get contraction. Okay, so EC coupling, action potential, activate the DHP receptors, and in turn activate the rhyodine to open up the channels, the calcium channels on the sarcoplasmic reticulum. Now calcium rushes out, and where does calcium go? Our calcium goes and binds to our troponin, moving tropomyosin, and now we can get contraction. We can get those heads binding. So that is the EC coupling. All those events that lead up to where we finally get contraction. All right. What the cue is, we go from excitation, get our action potential. Our action potentials are essentially our cue to release our calcium, and now we get calcium to initiate the contraction phase of the muscle contraction. So now we've released calcium, we get contraction. How do we get relaxation? We want to be able to contract the muscle, but we won't want it staying contracted all the time. We want to be able to relax it. So how do we do that? Our cue to get contraction is to flood the muscle with calcium. Now let's remove calcium, and if we remove calcium, we can't get myosin to interact with actin, and we'll get relaxation of the muscle. So we have pumps on the sarcoplasmic reticulum that sequester, pump back the calcium that was released through those ryanodines. So we pump back. And just keep this in mind, what do you think is a faster phase? Opening of the calcium channels and letting calcium rush out, or pumping it back? Yeah, the pumping back is slower. The faster is what's taking place here is very quick. This is slower, the pumping part. And therefore we're going to see contraction can happen very quickly, but relaxation is usually a little bit more drawn out. All right. And so here is that release. Here is that what's taking place, that action potential activates those DHP that are coupled. You see them coupled here with the ryanodine. And then that cues the opening of calcium to get our contraction phase. So our contraction involves calcium getting out there. We get the calcium, that's what the EC coupling gets our calcium out, allows for us to get contraction, and then we need to get relaxation. We want the muscle to contract, but we don't want it to stay contracted. We want it to be able to relax. So contract and relax. What do we get for contraction? We need calcium out there. Now, if we remove the calcium, we can get relaxation. Okay, and that's what's happening at the sarcoplasmic reticulum. There are these calcium ATPase pumps that are going to pump the calcium back in. All right, let me get my big head out of the way. And so when we do this, you can see when we get muscle contraction, we get boom, contraction. And one thing I want you to notice here is, what do you think is quicker? Is calcium being released by the channels, calcium rushing out, or is physically pumping the calcium back in? The rushing out is much quicker than the pumping back in. And so you'll see when we do a muscle contraction, we get contraction phase, which is quick, and the relaxation phase is more drawn out because this process here is slower. But you can see how much ATP we use for this muscle contraction, in that we use ATP to do power stroke. All right, so we use a ton of ATP to do that power stroke of the myosin head, and we also use ATP to pump the calcium back in to get relaxation. So we got to have calcium for both contraction and relaxation. So there's a state where our muscles stay when we die, pass away, they stay in rigor mortis. Those of you that have watched maybe some murder or law shows have heard of when rigor mortis sets in, and this is a state in which the muscles are staying contracted. And so think ATP wise why this might happen. We saw ATP needs calcium to be pumped back in, so calcium's out there so our myosin heads can bind to actin. And what else is if you remember the contractile cycle, when myosin binds and power strokes, until we get a new molecule of ATP bound to myosin, it will not release from the actin. So without ATP, which happens when we stop living, we stop producing ATP, myosin heads become attached to actin and won't let go, and therefore we get the state of rigor mortis. Eventually, the muscle just starts breaking down in general, the proteins start breaking down, and then the muscle will relax. Okay, but rigor mortis, lack of ATP, keeps us locked in. We also have our calcium that is available out there. Okay, so we have calcium, but lack of ATP, we won't be able to let go of, the myosin won't be able to let go of the actin. So let's look at what happens electrically to cause our muscle contraction. What are the steps involved, or what are the electrical activity leading up to the actual physical contraction? I'll get my big head out of the way here, but remember our neuron has to release neurotransmitter to start the action potential on the muscle fiber. So here is the neuron action potential recorded here, and you can see there's a delay between this action potential and that of the muscle action potential. Okay, because what do we have to do? We have to release that neurotransmitter, bind to nicotinic receptors, and then that induces. So those steps take a small step at a time where there's a delay between these and there. But what I really want you to focus on is the delay between this action potential taking place and just ending here, and till we see the actual force being produced in the muscle, which we see here. Right, and hopefully this is a little not correct in that what we say about the relaxation phase. The relaxation phase should have been drawn out much longer here. Okay, but let's look at this difference in time between here and then the actual force starting. There's a delay here. The other thing to remember about, if you remember from our neuro days, is that there is an absolute refractory period for these action potentials. Just as we have a refractory period for our neuron, we have an absolute refractory period for our muscle fiber. But look at here. I'll be out of absolute refractory period for my muscle fiber, and so I can induce a new action potential here. Right? I'm going to induce a new action potential here. What do you see happening with the muscle? The muscle, I can zap or give an action potential to the muscle several times before I even get into relaxation. And what am I doing every time I do that action potential? Every time I do that action potential, I'm opening up those calcium channels. So if I keep opening up these channels, letting more and more calcium out before I can even start relaxing, start pumping back in, what's going to happen to my calcium levels in the cytoplasm of my muscle? yeah, they're going to go up because I keep opening the channels. Calcium release, calcium release, calcium release. I keep opening them before I can even start pumping. That calcium is going to go up. And what we're going to see with this is that as we increase calcium, we're going to be able to reveal more actin. And that means if we reveal more actin with more calcium, we can get more myosin head binding. And what causes our force? Our ability, the more ability to cross bridge between actin and myosin will allow us to increase our force. So as we move along here, let's look at, as we stimulate the muscle quicker and quicker, we're increasing the calcium level, and we're going to see we're going to be increasing the force. Okay, but first let's completely go through this initial just one muscle contraction, which is a twitch, consisting of a contraction phase followed by a relaxation phase. And then we'll move into getting the muscle shocked more frequently. Alright, so here's our muscle twitch. And I'll get my big head out of the way again since it's sitting right here. And we have that latent period. So there's a, say our muscle action potential is here. We have this latent period in between my action potential and my actual contraction. And what's happening there are all those EC coupling events. Remember, our action potential cues are goes down those T tubules, causes the release of calcium. Calcium has to bind to troponin, move tropomyosin before this contraction actually starts to take place. So all those EC coupling events going from the action potential to getting to contraction are taking place in that latent period. But once we have that calcium and we've moved that tropomyosin, now we can start our contraction phase. Right? And what's happening? Contraction phase, and then after the muscle. So this is if we just stimulated the muscle once. We got it excited once, we get contraction followed by relaxation, because that pumping of the calcium back into the sarcoplasm, this should be more drawn out, and that gives us our muscle twitch. If we just were to give it one stimulation, this is what we would get. Contraction phase followed by relaxation phase. All right, let's look what happens when we start to stimulate the muscle more frequently, give it more frequency of stimulation. Okay, we're going to get summation. All right, we'll see this in a bit, and then we'll go into what's called tetanus. And we can go into incomplete followed by complete tetanus. And so we'll look at these and what's taking place on these diagrams here. All right, at each of these little arrows is I've basically zapped the muscle or zapped the neuron to get a action potential. And what we see, if I zap it at a certain frequency, I give it enough time. Here I'm giving the muscle enough time. I've excited the muscle, got it in the stimulus to cause a contraction. I get contraction, and my stimuluses are far enough in between that I get full relaxation. This is your muscle twitch. Now, if I start to increase the frequency, you can see I go from here to a much quicker stimulation. What happens? Now I'm opening the channels. Every time I activate and give it a stimulus, I'm opening those calcium channels. Now I'm opening the calcium channels here before I can get all the calcium pumped back. I give the calcium starting to pump back a little bit, but boom, I open the channels again and more calcium gets back out. So during summation, we're not getting back to baseline. The baseline is further up because I'm opening those channels before I can get all that calcium back into the sarcoplasmic reticulum. Right? If I start ramping up and give more and more stimulations, as you see here, okay, open the channels, open the calcium channels, open the calcium channels. So opening those calcium channels, I'm starting to flood the muscle with more and more calcium. The calcium levels are going up. Remember, more calcium levels, more revealing of actin, more cross-bridging, and that cross-bridging and that power between myosin and actin allows me to increase the force. And so as you see the stimulation going up, you see the amount of tension, the force that I can produce is going up. And as I increase it, you'll see at one point there will be a plateauing. But you can see we're still giving the muscle, it's not as frequent where we're getting this. There's still some time for relaxation. We're still giving the muscle a little bit of time to pump back a little bit of calcium. This is unfused tetanus. All right, and then I'll get my big head out of here. When I start to really frequently stimulate the muscle, all right, you'll see we get what's called the fused tetanus. Now I'm opening these channels so fast that I'm giving the muscle no time to pump back any of the calcium. And this is where you're going to find your greatest tension, because what do you find here calcium-wise? You have the highest amount of calcium because we just are flooding the muscle with calcium because we're continuously opening the channels, letting that calcium flow out from that sarcoplasmic reticulum. Okay. So again, what's happening here as we increase the frequency, we're increasing the amount of calcium, and that's increasing the amount of tension. Okay. So the calcium allows us more actin to be revealed, causing more cross-bridging, and therefore giving us greater tension. Okay. But again, just to review, why are we allowed to be able to create this tetanus is due to that short refractory period we see here. As I can continually open up the channels, boom, give an action potential, open up the channels, boom, open up the channels. Okay, and that'll give me where I get tetanus. I'm not relaxing, I'm getting no relaxation. Kind of make this point now before we get into cardiac is, would you want your heart muscles to be contracted into tetanus? No, because remember, our heart, we have to relax, fill with blood, then contract to pump the blood. So we're going to see in our cardiac muscles, we're going to see there is this longer drawn out action potential, and that won't allow us to do tetanus because we can't keep opening the channels. But here we can see we can zap the muscle fiber before we even get relaxation, so we can continually open these channels to flood that muscle with calcium. Okay, so it's that short refractory period that allows us to get that ability to summate and then get to tetanus. So now that we've been able to get our muscles contracted, let's look at whole muscles. All right, we're looking at individual muscle fibers on how we stimulate them. Let's look at whole muscles, what type of contractions take place with our muscles of our body, and then we'll look at some of the ways in which we can change the force produced by the muscles, our whole muscles. All right, And so type of muscle contractions, there is a type called isotonic contraction. And in isotonic, we're going to generate movement. We're going to get changing of the length of the muscle, all right? And we're going to create a force to move a load. There are two types. There is concentric and eccentric. Okay, concentric, think of, say I go to the gym and I'm going to do my biceps. Boom, I contract. Okay, that's concentric. But when I go to bring it down, my muscle is still contracted, but my muscle is lengthening. Right? That is an eccentric contraction. Okay, movement is taking place. Whereas with an isometric, a static contraction, say I go to lift a 50-pound dumbbell and I can't lift it. I can't create enough force to lift it. My muscles are still contracted, but I am not creating movement. I'm not creating force to move the load. And that is an isometric. So that's kind of seen here, and this would be isotonic, in which the muscle overcomes the load and we get contraction, or we get shortening in movement of the load. And isotonic here, the muscle's contracted, but we're not able to overcome the load, and therefore I'm not creating movement. The muscle is not getting shortened. And so why is this? How can we have contraction but not shortening of the muscle? Remember, there are two elements to, there's the contractile components, but there's also the connective tissue component, the elastic component of the muscle. So here's how we see we can get these different types of contractions, these isotonic and isometric, in that besides the contractile components of the muscle, there is the elastic components. Because remember, we're changing the size of the muscle when we're doing those concentric contractions, those isotonic, whereas the muscle length isn't changing when we're doing the isometric. But when I'm trying to lift that 50-pound dumbbell, I can definitely feel my muscles are contracting. I'm just not getting movement. And why is that taking place? Is because my muscles are made up of the contractile components, but also the elastic components. So you can see what's happening for isometric contractions is you can see between here to here there is some overlapping. You can see there's some movement of the contractile components, but what's happening is all I am doing is stretching the elastic elements of the muscle here, going from there to there. Therefore, the muscle is not changing size, but I am contracting the muscle. Whereas when I actually produce movement, I produce a concentric movement. You can see I continue moving the contractile elements. I already have, once I've done the elastic stretch, I can continue on, and now I'm shortening the muscle and creating movement and contraction. Okay, so in isometric, I'm only stretching the elastic component. In isotonic, I'm stretching the elastic component, but then further contracting the muscle to get the movement. Right, so we're going to move into looking at how muscles create these graded responses or graded contractions. I create greater forces, but to get there, we kind of have to talk about muscle subtypes. Okay, there are muscle subtypes. You probably have heard of fast twitch, slow twitch muscles, and you can see there are subtypes where speed of contraction and also resistance to fatigue. And you can see tension. Some muscles produce tension very quickly, some kind of in between, and some slower. So what do you think? A would be fast twitch, C, slow twitch. Okay, and so you'll see the three subtypes. A is this fast twitch glycolytic fibers. Glycolytic, going back to your favorite probably cellular respiration days, these muscles only do glycolysis, so they can work in anaerobic means. They don't have to have oxygen. They break down glucose, but you can imagine they can only produce 2 ATP by doing glycolysis. We'll see they are easily fatigued because they can only do glycolysis. All right, then on the other end is the slow twitch oxidative. Oxidative meaning, what are they doing? Oxidative phosphorylation. They're doing aerobic respiration, so they need oxygen to be able to operate. You can see they are much slower in their response. That's C here. All right, but we'll see they are fatigue resistance. All right. The fast twitch oxidative glycolytic, these are kind of the in-betweeners, the B. All right, and we're going to see these are kind of the muscles that we are training when we're doing differing types of activities, whether we're doing very powerful movements, very quick powerful movements like a power lifter, or if we're doing marathons where we need resistance to fatigue. We can kind of move these B's maybe closer to C or closer to A, depending on what kind of training we do. Okay, they are also labeled, you'll see 2X for the fast twitch glycolytic, 2A for the fast twitch oxidative, and type 1 for the slow twitch. Okay, so let's look at some of the properties, some of the characteristics of these muscles to get an idea of what we're looking at for these muscle subtypes. So muscle subtype and structure. Let's look at diameter. Imagine bigger diameter muscles are going to be more forceful. So your fast twitch are thicker in diameter. Your medium and then your slow twitch are smaller in diameter. Okay, what about myoglobin? Myoglobin is like hemoglobin. It is a source of oxygen, that is a store of oxygen basically in your muscles. If you remember our fast twitch, what do? They do glycolytic. So our 2X only do glycolysis. Do they need oxygen? They don't. So you can see they are the lowest of myoglobin content because they don't need oxygen to break down ATP to fuel the muscle. All right, your type 1 though are completely oxidative, and so they need that oxygen, so they have a higher myoglobin store. And then our type 2A are kind of the in-betweeners. They have some because they do oxidative, but they also do glycolytic. How about capillary density? Supplying blood. Again, blood's going to have the hemoglobin, it's going to have the oxygen, so we definitely need a high capillary density to type 1 that are solely doing oxidative. Type 2, we don't have to have a grand capillary density because they are doing glycolytic. We don't need to supply as much oxygen. And type 2A, again, are the in-betweeners. Okay, we already saw this on the one is fast twitch, much faster in developing tension, right? The tension's boom. Whereas for the slow twitch, the tension is much more drawn out. And what determines this is those myosin heads. The myosins are using the ATP. There are different forms of ATPases. The cleaving of that ATP. If I can cleave ATP faster, I can use it to do the power stroke faster. And so fast twitch are able to have ATPase isoforms that allow us to cleave and allow us to create this tension much faster. So fast twitch are fast, have a shorter, so they're boom, done. The slow twitch are much more slow and stay contracted for a longer period of time, but not as quickly as the fast twitch. Okay. So because the fast twitch are boom up and down, you can imagine what causes this relaxation is, right? Our calcium. All right, the fast twitch are able to pump the calcium back into the sarcoplasm much quicker. So we're able to get these rapid movements with our fast twitch. So looking at this kind of the cheat sheet, I don't need you to recognize, I'll pinpoint some of the things I'd like you to recognize. And where is speed of development of tension? Contraction duration, metabolism, right? And the other I want you to recognize is endurance. Okay, because these fast twitch, they only do glycolytic. So they're going to go into lactic acid cycle much more quickly. So these muscles are easily fatigued. All right, we can only keep up so much. Like we can't sprint. When we're sprinting, we're using fast twitch. Can we sprint for miles? No, we can't, because the muscles we're using for sprinting are fast twitch and they are easily fatigued. Whereas our slow twitch, okay, slow twitch doing a marathon, okay, they are fatigue resistance, or postural muscles, or we don't lag with our posture because those muscles, those slow twitch, are fatigued. And then our 2A are kind of in between the two. They are fatigue resistant, but not as much as the slow twitch and much more fatigue resistance compared to the fast twitch. Okay. But again, these are the ones that we can train. If we're doing marathons, we're training our muscles for marathons, we're moving these muscles to be more similar to the slow twitch. Whereas if we're doing powerlifting exercises, we're moving these 2A to be more similar to the 2X. Okay, so they're kind of the ones we can train into a certain direction depending on what kind of activity or training activity we're doing. All right, so let's get into strength and muscle contraction. Our muscles we have multiple muscle fibers and multiple types of muscles in each of our muscles. And so this can help us in determining what our strength of contraction is. And we can change the strength of contraction by the number of fibers we stimulate, how many muscle fibers we're activating, can change the amount of tension we produce, the thickness of the fibers. If I weight lift for a long time, I'm creating thicker fibers. I can create more force with those thicker muscle fibers. And then also the starting length. Where are they stretched? How stretched are my muscle fibers? And what orientation the sarcomere is can determine the strength of my muscle contraction. Okay, so we'll look through these three and look at their effects on how they affect the amount of tension I can produce. All right, so our whole muscles, we're going to use at how can we create different tensions, as our muscle fibers are made up, or sorry, not our muscle fibers, our whole muscles of our body are made up of many different muscle fibers, and they're arranged in what we call motor units. So we have multiple motor units that make up our muscle. All right, and this is the basic unit of a contraction in the intact muscle, and basically it consists of one somatic motor neuron and the muscle fiber that motor neuron innervates. Okay, so the motor neuron comes down, it just doesn't innervate one muscle fiber, it can innervate many different muscle fibers. And so when that motor neuron has its action potential, it's going to activate many different muscle fibers. Right? Each muscle fiber is innervated by only a single neuron. So get my big head out of here, in that these purple muscle fiber here are only innervated by this purple motor neuron. All right, the purple muscle fibers will not also be innervated by purple and blue. It's only innervated, the muscle fibers are only innervated by one motor neuron. And when that motor neuron activates, it's going to cause contraction of all. So when this purple motor neuron fires, it's going to cause contraction in all of these purple motor fibers, the ones that it's innervating. And what we're going to see, we have many different muscles, motor units. We're going to see some of these motor units can consist of large amount of muscle fibers or just small amount of muscle fibers. Where I want large, I want my motor neuron to be innervating many muscle fibers, is where I want gross motor movement or very forceful motor movements, such as my legs and my arms. I want to create forces, great forces very easily. I don't need this fine dexterity, but I got to get force. Then I would have motor units that have many muscle fibers in them. Whereas where I need fine motor movements, say as my fingers, all right, I need to activate different motor units very quickly. I would have very small amount of motor fibers in the motor units that control the muscles in my hand. Okay. So continuing on, what we'll also see, remember our motor neuron only innervates the muscles. All right, only one motor neuron can innervate a certain muscle fiber, and what we're going to see is a single motor unit always contains the same type of fibers. Okay, so going back to that last slide, the purple neuron, right, those muscle fibers it's innervating would be, say, all fast twitch muscle fibers. It wouldn't innervate some fast twitch, some slow twitch, some medium, some of the 2A fibers. It's going to be all fast twitch or all slow twitch for that one motor neuron. And as we said, the muscles are composed of multiple motor units of different types. And so the force of contraction, how much force I can produce, depends on the types and the number of motor units that I am contracting. So how can I vary my contraction? How can I vary between lifting a 10-pound dumbbell and lifting a 30-pound dumbbell? Okay, I can change the types of motor units I activate. Okay, I can start activating, when I do the 30, I can start activating more fast twitch muscle fibers. Okay, because they are more forceful. They create a greater force of contraction and more quickly. And I can also change the number of motor units that are responding. So when I do 10-pound dumbbell, I'm only activating a small amount of motor units, Whereas when I do the 30-pound dumbbell, I'm activating many more motor units to fire, and therefore I get more muscle fibers to contract. Okay, this phenomenon of increasing the amount of motor units activated is termed recruitment. I'm recruiting more motor units to activate, and therefore I get greater force of contraction because there are more muscle fibers contracting when I do recruitment of more motor units. Okay, so that is one way in which we can change the force. We can get a graded force produced in our whole muscles. So as increasing or recruiting more motor units, we can also train our muscle fibers to be bigger. So if we have bigger muscle fibers, we can increase the force. And so when we do some such stuff as resistance training, we do weightlifting, we're going to increase the size of those muscle fibers. Okay, so then when I go to contract or go to activate those muscle fibers, they're going to have more contractile components to it, more of the actin and myosin, and therefore I'm going to be able to increase the tension. Okay, how do we do this? We don't increase muscle fibers. We don't just increase the amount of cells. What we're doing is we'll increase the amount of actin and myosin in those myofibrils. Okay, more actin and myosin, we increase the size of the muscle fiber. Okay, if we do enough training, enough muscle training, all right, we can actually increase the amount of myofibrils. So first we start putting down more actin and myosin. If those myofibrils get too big, we can actually increase and create more myofibrils in the muscle. But what we're doing is increasing the size of the muscle fiber. We're not increasing the amount of the fibers. Okay, so size, not amount, is how we increase the size of our muscles. So when I go on lifting and my muscles get bigger, I'm not making new muscle cells. I am just increasing the size of the cells that I have. Okay, and first it's actin myosin, and then we can induce even myofibrils to be increased in the muscle. Okay, and so if I increase the size of those muscles, when the motor units fire for that muscle, I'm going to be able to create greater force. And then finally, how else can I change the force of the muscle? This is what we call the length-tension relationship. And what we're seeing here is that how stretched or slacked my sarcomeres. So again, my muscles are made up of the sarcomeres. I can stretch them, I can slack them, all right, depending on where I have my muscles. There is an optimal length of that sarcomere. There's an optimal amount of stretch that will give me my optimal amount of force to be produced. All right, and that is what we're seeing here in this graph. Here is the tension going up, and where do we see the tension the greatest? With the arrangement of C. Okay, the optimal resting length to create the greatest amount of tension in the muscle. And what we're seeing is there is a good amount of overlap between the myosin and actin. Because remember, what this is basically telling us is the ability to cross bridge is going to control the amount of tension that can be produced. Okay, in C, we're optimally linked. We have the optimum amount of interaction, ability to interact between actin and myosin here, and we're going to get the greatest force. When I go to stretch the muscle as we're going in this direction here, you can see there is less ability of myosin and actin to interact, and therefore the force starts to decrease as I further increase the stretch on the sarcomere. Then when I slack, what happens when I slack is they get thicker and they actually bulge out, and as they bulge, there is less ability for my myosin and actin to interact, and there's less room for it to move, and therefore my decrease again when I go in that direction. Okay, so I need to have that optimal stretch, because here is where I have the greatest ability to cross bridge. Okay, the tension produced is directly proportional to the number of cross bridges that can be formed. And C is where that's going to happen. There's the greatest ability to cross bridge or cross bridging, and therefore the ability to form the greatest tension. All right, so that's going to end it for skeletal muscle. As I said, most of this lecture is going to be on skeletal muscle, but we are going to dive just briefly into cardiac muscle and then into smooth muscle for these next few slides. And then again, we will touch on these cardiac and smooth muscle along the way when we get to the different organ systems in which they are utilized. All right, so here we'll take a brief glance into the cardiac muscle, and as you can see, it's similar in that there are these striations. So the contractile components are set up in sarcomeres, just as we saw with the skeletal muscle. The muscles look a little bit different in that they are small, little branched out muscle fibers, all right, and they are attached via these intercalated discs. Okay, we're going to see the intercalated discs. What do we have there? We have desmosins to tie them together because we're creating this force. But the one thing that we'll talk about later, but we'll look at briefly here, is the gap junctions. Because what we're going to see is through these intercalated discs, there are those gap junctions in which we can move a depolarization from one muscle cell into the next muscle cell. And this is going to allow us to depolarize basically all those cardiac muscles in a very similar manner, almost spontaneously together. Okay, but what we want to look at here is we're going to look at the EC coupling phase. Okay, very similar to what we saw in skeletal muscle. Remember, it is a sarcomere, so it has tropomyosin and troponin. So what we need to do is we need to cue the calcium release from the sarcoplasmic reticulum. Okay, almost similar setup through here in that we have the voltage-gated sodium channels bringing down the action potential. But when we get to the T tubules here, there is no DHP and ryanodine receptors. Basically, there are voltage-gated calcium channels, which are going to allow calcium to move in. Right. This extracellular calcium moving in is going to cue the release of the calcium from the sarcoplasmic reticulum to allow us to bind to troponin to move the tropomyosin to get contraction. This is called calcium-induced calcium release because the extracellular calcium coming in is inducing the opening of these calcium release channels from the sarcoplasmic reticulum. So this is the one main difference, is we have these voltage-gated calcium channels inducing the calcium release rather than the DHP and ryanodine that we saw on the skeletal muscle. Okay, but then once we have that calcium out, the contraction is similar because we have the same sarcomere setup as we do in the skeletal muscle. Now the oddball of the bunch is going to be our smooth muscle. There are no sarcomeres. Okay, they are arranged in this oblique type manner. You can see the sarcomere here, so there's no striations. We don't see striations in smooth muscle. That's why it looks smooth, because there's no striations there. But you can see this oblique pattern, this sarcomere, or sorry, not sarcomere, then this is going to walk in this direction, this one's going to walk in this direction, and you can see we get this contraction of this muscle. I like to call it like the booger, because it looks like a booger once it's contracted, but that is this oblique manner. It's going to give us this contraction, not just straight linear, but we're going to get this oblique type contraction. But again, you can see we get from this size muscle down to this size muscle. Okay, it is an oddball too, because we're not arranged in sarcomeres. What's controlling the contraction is not tropomyosin and troponin. All right, we're going to see a different way in which we're controlling the contraction of these muscles. All right, but first we always have to get them activated. And we're going to see there are two separate setups. Remember, the smooth muscles are controlled by the autonomic nervous system. And the autonomic nervous system releases via varicosities. And so there are two setups of this muscle we can have. Okay, single unit, right? And you can see there are these gap junctions. And the gap junctions allow, if this cell is depolarized, I can get depolarization of this one. When this is depolarized, I can move into there. So I only have to depolarize a certain amount of cells. Because of these gap junctions, I can move the depolarization through to these others. So I can run this axon via here, because if I get these activated, these muscles here, I can move that depolarization into the rest of the cells, surrounding cells. Whereas in the multi-unit, this is the less common. Most of them are set up this way. In the single unit, in the multi-unit, is I have to run these through because there are no gap junctions. I have to individually activate each muscle fiber. I have to depolarize each. So I have to release neurotransmitter near all of the muscle fibers, where with the single unit I can just activate these superficial ones, and the depolarization can move through the gap junctions to the remaining. All right. But once I have my muscle cells activated, let's look at the EC coupling. And this is where it gets a little bit, or a lot different from that of the skeletal muscle and the cardiac muscle. Remember, the cardiac and skeletal muscles are controlled by troponin and tropomyosin. Okay. The EC coupling in smooth muscle. All right, there is no troponin and tropomyosin, so there is no binding of calcium to troponin. What happens with our smooth muscle? All right, there are voltage-gated calcium channels. The depolarization, the activation, excitation of the muscle cells opens these voltage-gated calcium channels, which is going to bring calcium in. Calcium is going to bind to calmodulin. All right, and we have two sets of enzymes here. We have MLCK is myosin light chain kinase, this here. Right, it's a kinase, so it's going to phosphorylate the myosin light chain. All right, we have another enzyme, myosin phosphatase, or myosin light chain phosphatase. So we have one, the kinase is going to add a phosphate to the myosin light chain, and the phosphatase is going to remove the phosphate. So what gets activated by this calcium calmodulin complex. Remember that. The depolarization, the action potential, is going to open the voltage gated channels. That's going to activate my myosin light chain kinase, which is going to take the myosin that's not phosphorylated and phosphorylate the myosin light chain. This is the active form of myosin, and this phosphorylation of the light chain will allow the myosin to cross bridge and get activation and contraction of the myosin. When calcium levels go down, go lower, this opposite protein, the myosin light chain phosphatase, becomes activated. And it's going to do the opposite. It's going to remove this phosphate. And when the myosin light chain is not phosphorylated, the myosin head will not bind. Okay, unphosphorylated light chain will not bind. Okay, when it's phosphorylated, that's when I get binding, cross-bridging, and get activation. Okay, so this is how we're going to control contraction and relaxation. Not troponin, not tropomyosin. It's by either phosphorylating the light chain to cause contraction or dephosphorylating, or using the phosphatase to dephosphorylate to remove the phosphate and cause relaxation. All right, a little oddball. Again, we'll see this cardiac, we'll see the smooth muscle later on in the semesters when we're looking at the different organ systems, specifically cardiac, that we're going to be heading into here shortly. And then in digestive, we'll see some of the smooth muscle contraction and also cardiac for blood vessels with the smooth muscle. All right.